And now we will have Kate Adamala presenting on the biology on sample size of more than one. Kate Adamala is a faculty on University of Minnesota, and she's also leading the Build a Cell initiative. And the floor is yours.
Thank you. Thank you so much. Thanks for having me here. I am really happy that I'm able to talk to this community and kind of try to talk about engineering synthetic cells in a context that we don't usually apply it in, which is the origin of life and trying to investigate the earliest evolution of life. Because when you think about it, synthetic cells are essentially the simplest possible living or barely living organism that you can imagine right now. And the part of the rationale where we're working in this field is to try to reconstitute those absolutely earliest stages of evolution. And so I'm really happy that I get to talk in the same session as Nicola, because cell division is one of the absolute biggest and still unsolved problems. So I'm really happy to see progress in this field. So before I get into the weeds of experimental details, I just want to give you a couple of minutes of a general overview of the rationale of why we're doing it, because what we're doing is objectively weird. We're trying to build a living cell from scratch. And you might ask, why do we even bother doing that if there is such an incredible variety of form and function of living organisms? Why would you bother making a new organism instead of just studying what's out there? So from the evolutionary point of view, or if you're a chemist and you're just looking at life as on the biochemical level, there really isn't that much variety. All life came from this one last universal common ancestor, and there really is a single form of life on Earth. When you look at the evolutionary tree of life, everything goes back to this one root, and everything uses the same biochemistry. So that's kind of why, that's the reason, that's the rationale of why a lot of the people in our field think essentially modern biology is boring, because you only have one life form. If you only have one life form, you can't really learn any general properties of life as a physical-chemical phenomena in the context of trying to study the history, the past, and the future of the life in the universe, because we only can look at the life on Earth. So this is what we're trying to do in a field of synthetic cells. We're trying to kind of climb out of that gravity well of evolution. We're asking, what else is out there? What can we build? What kind of life we can build to learn how life started on Earth, and also to learn how life could look like. So right now, when you look at the life on Earth, you only see one instance of life. You only see what's possible as a result of this one particular lineage of evolution. And so we're asking, what's the crazy stuff out there? What can we build? And biggest problem with studying life in general is that we only have this n equals 1. We only have one known life form right now. And we also know that it had to start simple. It had to start from simple chemicals, probiotically plausible chemicals that arranged themselves spontaneously into something more and more complex until it started resembling a living organism. And up until very recently, we only had two ways of studying that problem. We could look at those probiotic reactions, very simple sets of biochemical reactions that were self-assembling, building slightly more complex structures, but nowhere near complexity of a living cell. Or we could study the entire living cell, which is essentially a black box. It's incredibly complex, it's expensive to work with, and it's very difficult to draw any general properties from looking at this very complex living cell. So this is where we are. This is where my field is. We are building synthetic cells, which are intended to be this middle ground between simplest biochemical reactions and the complex living cell. And there are two ways you can approach this problem. You can go either building top-down or bottom-up, so basically building synthetic cells from non-living components until it starts to resemble life, or taking a complex living cell and making it simple. And each way, whichever way you choose to go, the end result is this so-called a synthetic cell. What is a synthetic cell? There's not really a good definition right now. And if you watched Nicola's talk just a few minutes ago, you can think it's a liposome. Some other people would argue it's a microfluidic biochemical system that's capable of protein expression. Others can argue it has to be a cluster, but there's basically as many definitions as people in the field. And in general, most people agree that a synthetic cell is something that is not coming from an unbroken line of life since the last universal common ancestor. This is the kind of a unifying definition. And synthetic cell is something that has not been alive by the time we start our experiments. And so to me, the best simple definition of a synthetic cell is something that's more than a sum of its parts. It's something that's still understandable and controllable, but we can actually look at every single molecule in that system. And that brings me to the reason that makes this field the most attractive to me, which is you can control every aspect of a synthetic cell. So from the point of view of astrobiology and looking at prebiotic evolution, looking at life elsewhere in the universe, you can control every aspect of a synthetic cell, which means you can design it to resemble a synthetic cell under a lot of different conditions. You can build it so that it can withstand pressure, salinity, pH, other properties of environments that are found elsewhere, outside of Earth, or perhaps very extremophile environments on Earth. So that controllability is what really lets us build kind of a toolbox of synthetic cells that lets us kind of expand this lineage, go beyond the single lineage of modern terrestrial cells. And when I talk about synthetic cells, I talk about liposomes that express proteins. So it's basically a phospholipid liposome with a DNA genome inside and membrane proteins on a membrane and protein expression systems. So ribosomes, tRNA, cell extract, well, most of the time we use cell-free protein expression system. All that encapsulated in a cell that's about a micron to 5 microns in size. So this is the chassis that we're working on. And not just my lab, but everyone in the field is really excited about the applications that you can get out of it. There is this incredible interplay between cells that you can build and what you can learn about building cells. So if you build a certain functionality, like for example, the talk we just heard in this session, cell division mechanisms. If we can build cell division mechanism in a synthetic cell context, then we'll be able to learn more about natural cell division, but we'll also be able to have a synthetic cell that divides. And that also goes for a lot of practical applications, not just for studying the origin of life, for astrobiology, for space pharmacy, for medicine, but also for very terrestrial applications that are very immediately applicable to our current problems. And that includes metabolic engineering, that includes diagnostics and therapy, that includes even things like biocomputing. People use synthetic cells as a chassis for building computers. So you might think this is all cool, but this is never going to happen. It's absolutely impossible to build a cell from scratch. Well, I want to show you something that kind of always makes me laugh, and that's a citation from a New York Times article in October of 1903, where someone basically said the efforts to build a flying machine heavier than air are basically worthless. You might as well not waste your time. There's no point building a flying machine heavier than air because you will never actually achieve that goal. And then 2 months later, that Wright brothers flew their first Wright flyer in North Carolina. What that shows you is when people say something is impossible, other people go and do it. And this is kind of the approach that the field of synthetic cell engineering had. People are saying, it will never work. You will never be able to build a living cell from scratch. And what we do is we say, we'll show you, we'll go out and actually do it. So now let me show you one project from my lab that gets us a step closer to actually being able to say, this is a synthetic cell that actually lives. And it also answers one of the questions that Nicola had at the end of his talk, which is how do we build evolution into this system? So what we have is we engineered a system that demonstrates a complete cell cycle and evolution in a synthetic cell. So we start with a liposome. As I said, for me, a synthetic cell is always a liposome because cells have membranes. So we have this liposome with a DNA genome, and that DNA genome is replicated. We're using a phage polymerase called Phi29 to replicate that synthetic cell genome. Now there is a gene expression. the DNA genome gets transcribed into mRNA and then expressed into proteins using cell-free protein expression system, so-called pure system. Then we have membrane protein expression. The membrane protein in our system is a modified alpha-chemolysin, which is a very well-behaved membrane pore protein, and it's modified with 2 tags that get then displayed on the surface of a liposome, and I'll show you in a moment why that's important. If we have membrane proteins, we can have nutrient uptake and waste removal, so we can maintain the exchange of nutrients with the environment of a synthetic cell. Then we need to feed a synthetic cell. So most of the time when people feed synthetic cells, it's a completely artificial process where you have to add something and trigger that fusion with another liposome. What we engineered here is a genetically encoded fusion system that allows our synthetic cell to fuse with those so-called feeder liposomes. And those feeder liposomes enable growth of both membrane and the inside, the lumen of the synthetic cell. So we grow by uptaking those little feeder liposomes. And this is where that modified alpha-chemolysin comes handy. Because it's modified with 2 different his tags, there is a his tag presented on the surface of our synthetic cell if it expresses the alpha-chemolysin protein. And then we have those feeder liposomes that are tagged with a nickel NTA moiety on the membrane. And those feeder liposomes fuse with the alpha-chemolysin tagged synthetic cell. And that gives you growth, that gives you nutrient uptake because the feeder liposomes bring in feeding proteins, bigger molecules. And then that also gives you membrane growth because the feeder liposomes bring lipids. Now that's the cool part. If growth is genetically encoded, then you can apply selection pressure to it. In our case, the growth is proportional to the amount of that alpha-chemolysin protein on the surface of a synthetic cell. So if we have more of that alpha-chemolysin, the synthetic cell grows more. If we have less, it grows less. And that enables competition, that enables natural selection. We have 2 populations of synthetic cells. One of them contains the alpha-chemolysin, that growth protein, under a very strong promoter. We call it T7 max. And the other one contains the same protein under a control of a weaker promoter, just called T7. And the ones that contain the stronger promoter grow more. And if they grow more, they end up with bigger vesicles after each growth cycle. And the ones that grow less end up smaller after the growth cycle. Now, the most artificial part of the system is division. Until Nicola gets his job done and gives us a spontaneous division, we have to divide manually. So that's what we do here. We divide manually by extrusion. So we divide by extruding those large grown liposomes through a polycarbonate filter membrane, and that gives us daughter vesicles. And those daughter vesicles, if they come from a cell that grew more, you get more daughter vesicles. And the one that grew less gives you less daughter vesicles. So the stronger gene for growth gives more offspring. And that gives you essentially a natural selection. If something grows better, it produces more offspring. And then at the end, we developed a so-called generation counter to make sure that every cell in our system goes through actual 5, in our case, it was 5 cycles of that complete cell cycle. We divide manually, and then to annoy our alien neighbors that are going to stop my internet again, I will not talk more about the experimental details of this. Just the one kind of a experimental detail that I really want to mention is that we do have a generation counter, so we can tell for sure that cells do undergo all of those cycles. So for example, when we say we go through 5 generations of growth and division, we have a generation counter, RNA-based generation counter, that actually confirms that there is 5 generations indeed in that daughter cell after 5 generations. For some reason, it's always a daughter cell. It's never son cells. When people talk about liposome division, they always call them daughters. Okay, so we have 5 generations of growth and division of a synthetic cell. We grow, we have competition. The competition is enabled by a genetically encoded growth mechanism. And then we divide it, still artificially. We're not able to do a division in any spontaneous way. That's the job for people that actually work on the cytoskeleton. But this is the mechanism that shows you can have natural selection and you can have this complete cell cycle. So why is this complete cell cycle coupled with growth, with evolution and division still not life? And to me, it's not life, because NASA definition of life says it has to be self-sustaining chemical system capable of Darwinian evolution. So our system is capable of Darwinian evolution. It grows, it divides, it can do natural selection, but it is not self-sustaining yet. So when I first used that argument, to me, it's clearly not living because it's not self-sustaining. Someone made a counter argument that really made me think, and I actually don't know what's the correct answer to that. No organism is truly self-sustaining. Because, for example, I myself, if you dropped me on the surface of Mars, I would be dead within seconds. I'm not able to sustain myself without the environment. And so it's always the question of where do you draw the line at the experimental intervention? Where do you draw the line at how much you can do to sustain life of your experimental system? For example, if you have cells in a Petri dish, if you don't passage those cells at any given time, they're gonna die. So I guess it all depends on where do you draw the line. To me, it has to self-replicate. That's kind of my personal threshold for life. But I know a lot of people have different personal definitions, and it's always kind of a fun discussion. All right, so I want to show that slide again because it shows you that once we build synthetic cells, we'll really be able to explore things that nature was not able to explore. We'll be able to look at possibilities of life. We'll be able to look at how life could possibly work under different environments, how life could work if you build it from different building blocks, and very practical applications, like in space medicine, for example. In space exploration, we'll be able to use synthetic cells as little biomanufacturing tools. And we'll also, that's kind of relevant to this community, we'll be able to use synthetic cells to look for life elsewhere. Because right now, when we're looking at life in general, we don't really know what are the boundary conditions for life. And that's what we can explore with synthetic cells. By making life in the lab, we're able to truly explore what conditions are necessary for carbon-based biochemistry to be able to actually live. And so that not only includes looking for life elsewhere in the solar system, looking for past life on Mars, or looking for life on Enceladus or on Europa, but also looking for shadow biosphere on Earth. The shadow biosphere, this kind of a possibility of life being in a remote environment somewhere on Earth, is something that people find really attractive and look at it and try to kind of explore possibilities of using synthetic cells to explore life under different physical-chemical conditions. And now how that relates to space exploration. When you have a synthetic cell, you can think of it as a little bioreactor. If you have a bioreactor, you can make molecules, you can make whatever you want. And if you have a synthetic cell, you can completely digitize the information that is needed to make a synthetic cell. That's the fascinating part for remote applications. I can send information about making my synthetic cell to someone in Poland right now. And if we already agreed previously on a set of building blocks, you should be able to make that synthetic cell in your lab today. And that's different than being able to ship actual biological samples, which requires actual physical transfer of materials. So that's obviously all fun if we want to do it right now, but I could send you a FedEx package that you can have tomorrow on your desk. The real interesting applications emerge when we go to the locations where FedEx doesn't deliver. And that includes long-term space missions, that includes potential colonies on the moon or on Mars. And so that kind of opens up possibilities of using synthetic cells as bioreactors, as ways to engineer molecules, as ways to make drugs, ways to make metabolic engineering remotely in places where you cannot normally ship things like reagents and supplies. So this kind of, I hope, convinced you that this field is growing really fast, and this field is really enabling a lot of applications, both in basic research and in basic foundational research and in practical applications. And I just want to plug our international synthetic cell community. There's a lot of us, people that believe that building synthetic cells will one day happen, is happening already, and is necessary for us to truly understand the nature of biological life. And we started this so-called Build a Cell organization, which is an open community of people that believe in engineering synthetic cells, believe in growing this field, growing it safely, and also growing it with all of the implications for the bioethics, biosafety, biosecurity. And I want to wrap up by saying thank you. Thank you to everyone who works in my lab. Thank you to all those amazing people that joined my lab and want to work on this relatively weird topic. I want to thank to all of you for bearing with me when my internet died and listening to me, and thank all our funding agencies. And thank you so much, and I will take questions.
Thank you, Kate. I had to take this part of session. My name is Mikołaj Kuska, because Tom is co-organizing Lynn Rothschild lecture. So we have 2 questions, if you can see, or
yep.
3. First question should be from Mark Elovitz.
Yep.
I didn't. Yes. So
I see it. Should I read it?
If you can, would be great.
Yeah. Okay. So Mark's asking, have you considered bombarding the synthetic cell with random high energy particles to try trigger mutations in the cell's genetic material? So funny that you ask. We actually just sent samples to someone who has access to 2 really cool devices. One is a planetary simulator. So it's basically a really fancy temperature controlled vacuum chamber where they simulate radiation and radiation temperature, pressure, and gases. And we're going to do tests of interstellar travel and Martian environment for seeing the mutation rates in synthetic cells. And we're also, the same guy has an access to the experimental reactor. And so he's going to subject our synthetic cells to the samples of hard radiation, and we'll see what happens. My guess is the mutation rates will increase significantly more than you see that in live cells, because we don't have any repair mechanisms. So if you cannot repair, obviously the mutation rates is going to skyrocket. So this is going to be, I'm really excited, and we should have results from those experiments in a few months. Okay, so next question. Yes, Nicola, vesicle is feminine in Italian. It's feminine in Polish too, and so I have no problem with calling them daughters. Okay, Tomasz is asking, those liposomes that grow less become smaller or still grow but less? Yes, they still grow, but they grow less. How does the RNA-based generation counter works? So RNA generation counter is based on a promiscuous ligase, activity of a promiscuous T7 ligase that is capable of ligating sticky ends of RNA. I know it's originally a DNA ligation enzyme, but it's been demonstrated by other groups before for the purpose of preparing libraries for deep sequencing that you can use that ligase to ligate RNA. So what we have is a double-stranded short oligos of RNA that creates sticky ends. And those sticky ends only can ligate in one very specific configuration. And with each generation of feeder liposomes, we're introducing 1/2 of each generation of those sticky ends. So only if a synthetic cell goes to 5 cycles of feeding, it's capable of making this what we call full-length ligation product. And we detect that product using qPCR, RT-qPCR. So only if we have all 5 building blocks from 5 rounds of growth and division, we're able to grow this full-length product. And that's what tells us that that synthetic cell went through 5 cycles of growth and division. And now, another question, alpha-chemolysin has his tag. His tag, what method do you use to detect his tags? So we obviously run Western blots, so we can tell it's definitely there. And we inserted it on 2 ends of the alpha-chemolysin. So it does display on a membrane regardless of what side the alpha-chemolysin inserted itself into the membrane. And the negative control was that we used alpha-chemolysin without that his tag, and there's no fusion. So we don't have a way to directly visualize the his tag on a surface of the liposome, but we can tell that the his tag is necessary for fusion. And then Nicola's asking, thank you, Kate, for a very nice talk. Thank you so much. I share many of the ideas you discussed. Question, do I understand correctly that you managed to replicate the pure system itself inside your daughter son cells. And no, the pure system does not replicate. The pure system renews itself in the feeding cycle. So with each feeding cycle, we're delivering fresh ribosomes, tRNA synthetases, and all of the components. But what you're asking is not out of the question, because we know that from the experiments of the Forster lab and Danelon's lab, that it's possible to replicate the entire plasmid, the megaplasmid that carries the pure system inside the synthetic cells, and that's using the Phi29 replicase. So it's possible to replicate the entire translation system. We settled on delivering it by feeding because we wanted to see how far we can go with generations, but it's also possible to just do it kind of in a single cycle. So I think I went through all of the questions. Thank you very much.
Yes, so thank you so much for your presentation. It was great. And that's all.
Thanks. Thanks, everyone. Bye.
Thank you. So I'm gonna close this session. Thank you.